So now we will be dealing with DNA sequencing process, okay? Sanger sequencing process. Now listen. Now when I am talking about the process, you should always understand that there are some intricate chemistry behind these things, okay? Now listen. We have already discussed that there are actually two types of nucleotides we are using. Number one, that is normal nucleotides or DNTPs. Number two is abnormal nucleotides or DDNTPs, dideoxynucleotide. Okay, now listen. What is the role of DDNTPs? Okay, now we will talk about this. Now listen. Suppose this is a DNA molecule. DNA is double stranded, right? Okay, now listen. Now for sequencing, what you will have to do is you will have to separate the strands using a thermocycler machine. That means both these strands are separated. Now listen, what you will have to do next is you will have to add one primer. Suppose this is the primer. Don't worry, I will talk about this in details. Don't worry, I am just talking about right now the role of DDNTPs. Suppose this is a primer. Okay, now first things first, suppose here it is A, here it is T, here it is G, here it is C. Suppose I have added DDNTP. Okay, now DDNTP, there are four types of DDNTPs. There are four types of, like there are four types of normal DNTPs. There can be four types of normal DNTPs. That is DATP, DTTP, DGTP, DCTP. Similarly, there are four types of DDNTP. DDATP, right? DDTTP, DDGTP, DDCTP, right? So now suppose I have added all these things here. I have added this DNTPs and DDNTPs here. Suppose now here what will be added? What? T, A. Suppose here a DDGTP suppose is added. Now then and there the reaction will stop. Why? Because for addition of another G, normal G, for addition of normal G here, CTP, right? Here should be CTP. Here should be what? C, no? Suppose this C is DDCTP. Suppose this C is DDCTP and it has come here. Suppose this DDCTP has come here. Okay? Then and there, no further nucleotides will be added in this particular section. Why? Because you remember that from your RNA polymerase lecture that, from your DNA polymerase lecture, you remember that DNA polymerase only extends the chain by realizing this OH group, and then it, what happens? Then another phosphate group which is present in another nucleotide okay. Now listen, this is actually the reaction between a nucleotide and another nucleotide, right? Now listen, if you don't have this OH bond, OH group here, that means no oxygen and phosphate bonding will be there. That means in DDNTPs, as instead of OH there is one H, that means whenever DDNTPs are incorporated by DNA polymerase, then and there this whole reaction will stop. Okay, this is the importance. If you can't understand how these things are working, go and revise DNA polymerase lecture, you will understand the basics. Okay, now listen. So this is the basic chemistry of how DDNTPs work. Now let's take an example and we will try to solve the sequence of this particular thing. Now listen, there is one important restriction enzyme by the name of EcoR1, right? There is a restriction enzyme by the name of EcoR1, right? Now everybody knows, suppose that EcoR1 is a restriction endonuclease, right? Is a restriction endonuclease. What do you mean by endonuclease? Endonuclease cuts the nucleic acid from middle portions, right? Not from end like exonucleases. Now listen, EcoR1 is an what? Endonuclease. That means what? That means you will have to understand that suppose this is a DNA sequence, endonuclease will always cut like this, not from end, right? Because they are not exonucleases. But listen with all your attention. Very basic point you need to understand about restriction endonuclease. This is a restriction endonuclease, okay? This is a restriction endonuclease. Now listen with all your attention. Why they are called restriction endonuclease? Because their endonuclease activity is restricted to a particular DNA sequence. That means EcoR1 will never cut here, 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 or anywhere like this here. Then where will DNA EcoR1 work? Listen, EcoR1, this enzyme knows only one sequence, that is a palindromic sequence, GATTC. What is a palindromic sequence? Listen, C, this will be G, A, T, C. Listen, read it from here. This is GATTC, right? I remember it as GATTC. Okay, read it from here. It is also GATTC, right? That means EcoR1 will try to find this gut, will try to find this stomach or gut, will always try to find this GATTC, okay, sequence. If it finds any GATTC sequence here, then only it will cut only here, not here, not here, not here. Okay. Now that's why they are called restriction endonucleases. That means they are endonucleases. They will cut from the middle, no doubt about this. But their restriction endonucleases means what? They are restricted only to a particular DNA sequence. Now listen. Suppose we are a very big, science corporate. Okay. That means we are holding a big laboratory and we are on the verge of discovering GATTC, this sequence, the sequence which is recognized by EcoR1. Suppose you are in a, parallel, universe, and that universe is not at all advanced with respect to ours, which is a joke, but still, okay. Suppose, just remember, imagine that in that particular, parallel universe, no one knows what is EcoR1 and no one knows GATTC. Now we want to sequence the GATTC. We want to know that which sequence is actually cut by EcoR1. Okay, we want to know which sequence is cut by EcoR1. Now listen, in this universe, not in the parallel universe, if you want to know, the best particular method is restriction mapping. Okay, but suppose in that universe we don't know even restriction mapping, we know Sanger sequencing only. Okay. Suppose we know in the parallel universe Sanger sequencing. Now listen. Let's start. Listen. Suppose this is a particular DNA sequence. Suppose this is a particular DNA sequence. Listen. Suppose this is a particular DNA sequence. Okay. and here it is. G A T T C, right? Get T C. Now listen. What will happen is, in the first step, what will happen is, listen. If you want to, if you want the sequence of the, suppose you don't know what is the exact sequence. That means I am writing in the board obviously, but we don't know. Suppose we are blind. We are blind towards this sequence. That means we don't know the exact sequence. We just want to discover this. Okay, now listen with all your attention. It's very simple. Okay, I will give you homework with respect to these things. It's very important. Now listen. Suppose you take this whole sequence, this whole DNA in a beaker. Suppose, okay, in a parallel universe. In a parallel universe, we are all full. Okay, now listen. So in the beaker, we will have to add this sequence, then we will have to add DNA polymerase, right? Then we will have to add magnesium sulfate, then we will have to add primers, but only one type, forward or backward. Suppose in a meeting, in a confidential meeting like this, we have decided that there are two, two strands of a DNA. Suppose in a confidential meeting we have decided that we will sequence this particular DNA strand. Then reading this we will understand this, okay? How to do this. Now listen. So we are assuming that we will be doing the sequencing of this. Both can be done. Yes, it's a laborious job. Okay? In the movie Titanic, they only build the half part of the ship. Right? It's very similar to that. They only build the half part of the sheet. Obviously, it was a sheet. Okay. Now listen. Whenever they wanted to show the full ship, they would just flip the ship. That is what we are using here. Yes, you should always know, in America, in any part, you will get many intelligent person. Okay, obviously. Now listen. They are using same concept here. What you have just now heard from me regarding Titanic, they are using the same concept here. Listen, what is that? We will just sequence one strand, then using a software or using our own hand at least. This is just a 6 nucleotide long sequence, so we can guess it, right? But if it is 600 long, yes, we can guess it. If it is 6,000 long, we can laboriously guess it. If it is 15,000 long, get out. Right? Now listen. That means, obviously, when the guessing part becomes more, then let us drop the sequence of one of the strand to computer. Let computer do whole work. Then we will take the sequence and we will upload it to the online library. Okay, that is our job. Suppose human genome sequence has still not hit in the parallel universe. We are living in a junk then. Okay. Now listen, at least with respect to biology. Listen. So suppose in a beaker, I don't know which PCR machine will take the beaker, but still, suppose in a beaker you have taken a DNA sequence of known sequence, and we will, for the sake of controversy, we are just saying that we don't know the sequence, but we can see it. I don't know how, but still, DNA polymerase, magnesium sulfate, and one primer, which will be ligated. Now listen, which will be ligated to this overhang sequence. This is called overhang. That means this is our area of interest. These are overhangs. Okay. So we must design the primer in such a way that the primer is attached to the 5' end of the overhang. Okay, not the 3' end. Okay. That means if we have chosen this particular path, we will have to start from here. Primers are always chosen from 5'. Okay. in computer when we design primers using softwares like primer 3, we always do this. Okay, we have a separate class. Okay, now listen. Now point you need to understand here is that we have already designed a primer, right? Suppose using primer 3. So we are grateful that in the parallel universe there is something called computer and we have primer 3. Now listen with all your attention. Suppose we want to sequence this particular portion. Okay, now listen, what we will have to do first, we will have to run the thermocycler. We will have to first run the thermocycler so that primers can anneal. We have already added the primers, but primers will never anneal. Suppose this is a double-stranded DNA and there are primers, okay, obviously, but primers will only attach to this single-stranded DNA when it is single-stranded. That means it will have to what? The double-stranded DNA will have to what? Denature first. For this denaturation we need heat, and this heating process is done by thermocycler. So successfully, thanks to God, our primer has annealed to single-stranded DNA. That means we are not considering this strand. Suppose, okay, this strand is not there. No, it is there, but this strand is of no use right now. Now listen, we have done this, right? Now suppose it is a 4 ml solution. 4 ml solution in a beaker. We are genius. Okay, 4 ml solution. And now listen, we are dividing this 4 ml solution into, we are taking this 4 ml solution into 4 what? 4 holes, 4 PCR tubes or Eppendorfs. Okay. Okay, now listen. So these are 4 tubes. Okay, now we have added this single-stranded DNA to every portion. Okay. Finally, we have broken the, beaker. Okay. Now listen. We have added 4, 4 ml, 1 ml to each. 1 ml of this what? Sequence to each. Now listen. So this is our. Now listen. Where is actually, this is called mix, okay, mustard mix. Now listen, where is actually the DDNTP? Where is DNTPs, right? Now listen, after we have divided this whole solution into equal parts, then after that we will start adding DATP, DTTP, DGTP, D what? CTP. Okay. Now listen, I have only added deoxyribonucleotides, Where is the dideoxynucleotides? Listen, in each, now listen with all your attention. This is the unique thing about this particular sequencing. Listen, if you don't understand this, you will never understand Sanger sequencing. Listen with all your attention. In each test tube, just add 1 DDNTP at a time. That means to this test tube, we'll add only one, that is DDATP. That means to each test tube, just add 1. 1 means not 1 microliter or something like that. Okay, but this concentration should be less than this. Okay. Now listen. To this we have, this is what? This DNA, single-stranded DNA, ligated with primer, polymerase, right? Now listen, I have done one mistake, that is polymerase should be added at last. Why? Whenever polymerase is added, it will start the reaction. But if there is no substrate, polymerase will not do anything. So listen, in any biochemistry reaction, always remember, in any biochemistry reaction like PCR, which involves an enzyme, the enzyme is added at last. Remember this for your entire life. Okay. Now listen. So, okay, let's change the let's change the look of this board right now, because we need an agarose gel. Okay. So, what is this? Single strand DNA, right? After that, D ATP D TTP D GTP D CTP. To this again, same thing is added again and again to each and every what? PCR tube, okay? But what is the unique thing? Listen, every test tube contains single stranded DNA. This is DNA, right? This is DNA. This is DNA, okay? Now listen. Now listen with all your attention. Now listen. So, now this test tubes contain single stranded DNA as well as all the 4 deoxyribonucleotides. Now listen. This is the first tube. This is suppose the first tube. This is suppose the second tube, and this is the third tube, and this is the fourth tube. Okay. Suppose. Now listen. Oh, not suppose. It is 1, 2, 3, 4. Okay. Now listen. Now listen. So to the first tube we are adding DDATP. Okay. To the second we are adding DDTTP. To the third we are adding DDGTP. To the fourth we are adding DDCTP. Okay. Now listen. Now listen. After we have mixed all these things, we will add magnesium sulfate, and then after we will add DNA polymerase. So, DNA polymerase, DNA pol, DNA pol, DNA pol, DNA pol, magnesium sulfate, magnesium sulfate, magnesium sulfate. Okay, now listen. Now, within seconds, this reaction is done, and you have sequenced the gene. You have sequenced this, DNA. Oops, oh my God, I have wrongly, what was the sequence? What was, obviously that is GAT tissue, but from here it is GAT tissue or from here? Actually, 3 prime, right, like this, right? Okay, that's it. This was the primer, right? Now listen with all your attention. Understand this very basic thing, okay? This portion you will have to sequence, right? You will have to sequence this portion, right? You will have to sequence this portion. You will have to portion this sequence. Okay. Okay. Now listen with all your attention. In the first test tube, we have added what? DDATP. That means what? Whenever, listen. Where are T present in this particular sequence? Here and here, right? Opposite to T, what will be, this will be G, A, T, T, what? C, right? This will be a sequence. Now listen with all your attention. Now listen. Where are T present? Here and here, right? This is a 5 prime. This is a 3 prime. Now listen. Now we will have to add all these mixtures in a acrylamide gel. okay, in an acrylamide gel. Clear? In an acrylamide gel. Now listen with all your attention. We will not, add these contents of this test tube randomly. What we will do is, we will add in this lane only A's. That means where DATP is poured. We'll add in this lane where DTTP is poured. We'll add in this lane where G is. Now listen. So now we are taking a micropipette. We are taking a micropipette and we are taking this and we are pouring it here. Okay. Now listen. What is the choice? What is the condition now? Listen. This was the sequence C T T A A G, right? This was the known sequence. Okay, now listen with all your attention possible. Now, DNA polymerase has already started working. That means, what will be incorporated here? Opposite C is G. Opposite T is A. Now listen, the third, again there will be A, right? Now suppose the third A is actually DDATP. Suppose the third one is DDATP now. That means after that, no nucleotides will be allowed. That means, now listen, that means, listen with all your attention. Suppose the length of this piece of nucleotide or the length of this piece of DNA is suppose 20. Suppose 20. Okay, let's make it 10. Don't worry, this is a regular practice. Okay, suppose this is 10. Now listen. Only 2 nucleotides, after 10 nucleotides, only 2 nucleotides are what? Incorporated now? That means, what will be this? There will be 10 plus 2, 10 is coming from what? This primer. That means 12 nucleotide long. The sequence of this particular portion is 12 nucleotide long, right? Right or wrong? No, it is 13. 1, 2, 3, 13. Okay, 13 nucleotide long. That means suppose you are writing here. So we have taken this from A, right? A test tube where DDATPs are produced. Suppose 13. 13 is a quite small thing, so it will come much more. What? Suppose this is the lane. This is the lane. Okay. This is the lane for running the materials. These are the lanes. Okay. Now listen. This is the lane. Okay. So this is 13 base pair. Now listen. Write it down. 13 base pair. During exam you should always write it down. Now listen. In the second cycle, what has happened is, in the second cycle, suppose what has happened in the second cycle, this is 10 nucleotide again. G A. Now listen, in the second cycle, what has happened is, the second A is actually DDATP. Okay. Then what is this? 10 plus 2, 12. Means here will be the band. This is 12 base pair. Okay. Why it is nearer to that? Because it is 12, it is 13. You know mathematics more better than me. Now listen. Let's talk about T. During exam, always write this, the known sequence. Okay. That means the sequence you are about to be sure of. Now listen. Now listen with all your attention. Suppose this is your primer. Now, so whenever T will be present, there is a chance of getting what? DDTPs. Now listen. G A A T. Suppose the second A is in place of second T. Suppose DDTP is incorporated. That means here the chain is terminated. That means 10 plus 1, 2, 3, 4, 5. That means 10 plus 5 is, oh my god, you are telling 50. 15, okay, good. You are educated. Okay, now listen. Okay, let me draw all these things with what? Let me draw all these things with red. Now listen, this is 15, right? So here it is 15 base pair. Suppose here this is 15 base pair. Now listen, you know that why when the base pair is increasing, it is becoming, you know, upper. It is the band is coming on upper side. Why? Because the you know, longer the length of the DNA, the more it will be restricted in the agarose gel. Okay, now important point to understand. What is the percentage of this agarose gel? This is run on 12% agarose gel. Okay, this is run on 12% agarose gel. Okay, with a power of 40 milliampere. 40 milliampere, and for 6 hours. Yes. Now listen with all your attention. You should always understand that this was actually the calculation of Friedrich Sanger. He actually used it. 12% agarose gel, 12% not agarose gel, sorry, not 12% agarose gel, acrylamide gel, polyacrylamide page, acrylamide gel. Okay. Okay. Now 40 milliampere of current and for 6 hours. Now listen, this is 15 ampere. Yes, sorry, this is 15 base pair. Now listen. In the second case, this is the primer. Again, what? G A A. Now in the second case, what we are getting? Suppose this T is what? DDTP. DDTP. Now listen. This is 10 plus 4, 14. That means what? Again a closed band. Okay. Let me go like this. It's closer to 13 and yeah, right. This is what? 14, right? Okay. Now listen with all your attention. Now let's come back in the next stage. Why? Because there is no what? T's left. Whatever you do, you will get only these two things. Whatever you will do, you will get only these two portions. Now let's talk about this. This is the sequence. Okay, what? CTTAAG. Okay, so this is your primer. Okay, this is your primer, and here it is what? Now listen, what we have added? DDGTP, that means opposite C is G, that means you should be aware of G's. Here it is G. Suppose, is there any option that G will be added in any other, you know, place? That means the first G will be what? Maybe the first G will be DDGTP, right? That means what? 10 plus 1, 11. In the G portion you will understand it is like this, lowermost position. Okay, this is what? 11. Now listen, during your exam you should always write like this, otherwise you are bound to get confused. You are bound to get confused. Okay, in any exam, if this is given and you are asked to find out the sequence of this thing, you should always remember the base pairs. Then you should plot these base pairs in this particular diagram, then you can find it out. I will be teaching you how to find out using these things later, just after 2 minutes. Now listen, let's talk about this, the last one. Now listen, CTTAAG. Are you laughing at yourself? Now, what is this? C, that means opposite G is C, that means what? G, A, T, T, C will be what? DDCTP, right? That means 10 plus 1, 2, 3, 4, 5, 6, 16, uppermost, 16 base pair. Is there any G available? No. Now listen, how to read this gel, right? How to read this gel, right? You now remember this for your entire life. In Sanger sequencing or dideoxy sequencing, always read from top to bottom. Yes, always read from top to bottom. And the lowermost portion represents 5 prime. Uppermost portion represents 3 prime. Now listen, draw this. Okay, this was your overhang. What is the most important? What is the first thing you have got? Sorry, this is your 3 prime. Sorry, this is your 3 prime and this is your 5 prime. The topmost is the 5 prime. Now listen, uppermost, what is the lowermost portion you have got? G or C? G, this is the 3 prime, okay? G. After that, 11, what is the lowest base pair after 11? 12, A. Okay? Then, lowermost after 12, 13, again A. Lowermost after 13, 14, T. Lowermost after 14, sorry, higher with respect to 14, 15, that means again T. Again, after 15 what comes? This 5 prime. You have got this sequence, right? You have got this sequence. Now don't write. Now listen. You will have to always understand that using Friedrich Sanger sequencing or dideoxy sequencing, you will always get that particular sequence for which you have used the primer. Okay, again another thing, the direction in which you are getting the sequence is opposite to the direction of primer, right? See, and now listen, you will always have to remember the lower portion is 3 prime, the upper portion is 5 prime. Okay, now when you have got the sequencing of one of the strand of DNA, you can easily guess what will be the strand of another, right? If this particular sequencing, if you are sequencing a long portion of DNA, then you will have to use a software for doing this thing. If you are only doing it for 100 or 200 nucleotides, you can easily do that, right? But it is really time consuming. It is really time consuming. Why? Because this PCR thing takes time. Okay, number one. Number two, you need an enormous amount of inner peace. Why? Because if you forget, suppose you are loading 100 of these things, if you forget, suppose you have loaded A here, but suppose for only one thing you have just loaded A in the T column, the entire thing is gone. The sequence will be what? One sequence will be unknown. Now listen, you can easily tell one sequence I can guess from 4. No, try permutation combination, then you will understand what are the possibilities. Because permutation combination is very, very easy when there is small number of small amount of DNA, but it becomes enormously large when the number of nucleotide increases, right? Okay, now listen with all your attention. So you have understood that how to sequence this, how to sequence a particular gene or how to sequence a particular DNA sequence using Sanger sequence, right? Now listen, we have understood what is Sanger sequencing right now. Now listen, when suppose this is a gene, okay, suppose this is a gene. Now few details about Sanger sequencing. Suppose this is a gene and suppose this is your sequence, okay? You want to know. Now listen, something called coding strand and non-coding strand? You know what is coding strand and what is non-coding strand? What do you mean by coding strand? The strand of the DNA from which mRNA is produced. Suppose this is the strand from which mRNA is produced is called coding strand, right? This is called coding strand. Then opposite to the coding strand will be non-coding strand, right? Now listen. Suppose this is the DNA. Suppose this is C, T, A, G, G, C, T, C, C. Okay. Suppose this is your strand from which mRNA will be produced, right? Now suppose this is your mRNA. Suppose this is the mRNA which is produced from, which is transcribed from this particular strand. Now what is the sequence then? What is the sequence then? G, A, U, C, C, G, A, G, G, right? Don't you think this strand looks similar to this strand? Right? This strand looks similar, apart from you, this strand looks similar to this, no? Right? That's why coding strand is called nonsense strand. That means it has no sense similarity. It has no similarity. No sense similarity means what? No similarity with respect to mRNA. But non-coding strands are called sense strand. Why? Because the mRNA actually looks like this. Okay, now listen with all your attention. Coding strand is also known by the name of template strand. Is also known by the name of template strand. Okay. Now listen, in many books you will find that non-coding strand is actually preferred for Sanger sequencing, and we actually put the template strand with respect to this. Okay. In many books you will find these terms, that's why I have told. You can find it in details if you revise your transcription chapter, which I have already read, which I have already taught. Obviously I have read many a times. So in many books you will find that you will have to, design the primer according to sense strand or non-coding strand, then you will have to guess the template strand, or you will have to put the sequence of sense strand into a computer software, and the computer software will give you the template strand. Okay. So this was the whole of Sanger sequencing. Okay. Next we will be talking about Maxson-Gilbert sequencing.